KC is a 24-year-old woman who arrived at the emergency room in agonizing pain. She was suffering from photophobia, an extreme physical aversion to light, and the moment doctors pulled her sunglasses off, she vomited all over the floor. Her neck was totally stiff, and her words were bleeding into nonsensical talk about her boyfriend. A needle pierced her lower back to collect cerebrospinal fluid, which is supposed to be clear. Hers came out cloudy and yellow. Something was living, eating, and reproducing inside her nervous system, causing meningoencephalitis, a severe inflammation of the brain tissue. Around the same time, JW, a 66-year-old grandmother, presented to a hospital with a bizarre emergency. She was baking cookies when her hands suddenly felt like they were on fire. Over just a few days, that intense burning throbbed down into her knees and ankles, making it nearly impossible to walk. Her primary care doctor initially suspected arthritis, but the resident treating JW noticed the pain was incredibly sudden and completely resisted steroid treatment. When arthritis appears this quickly and aggressively, it usually means a pathogen has physically invaded the joint space. He tested for hepatitis and Lyme disease, but both came back negative. Despite the social awkwardness of questioning a grandmother in a monogamous marriage about her private life, the resident looked at the final possibility on his list. Gonorrhea is a pathogen that typically stays at the site of contact, but in rare cases, it escapes into the blood and settles in the joints, forcing extreme inflammation as it grows on the bone. He ordered the test, and it came back positive. The resident remembered Casey's case from weeks earlier. She had also mentioned intense joint pain before her thinking became tangential. Doctors ran a specialized test on her yellow spinal fluid and found the exact same result. A common sexually transmitted infection was now swimming inside a young woman's brain. Under normal circumstances, Neisseria gonorrhea is a localized infection. It is a fragile bacteria that almost always remains isolated to the initial site of physical contact. That health officials in Michigan noticed a break from this baseline. In the 4 months leading up to August 2019, 16 different patients in the same region were hospitalized with disseminated infections, meaning the bacteria had successfully broken out of the initial site to attack the heart and joints. Genomic sequencing of these patients revealed a specific mutation called the PORB1A gene. This gene creates a specialized version of a protein that sits on the outer surface of the bacteria, changing how the pathogen interacts with the lining of human cells. This mutation alters the bacteria's behavior, allowing it to traverse in and out of human cells and move through various types of tissue. Instead of staying anchored at the infection site, the bacteria hitches a ride in the bloodstream to reach the brain and the joints. Doctors managed to identify Casey's infection just in time. They treated her with ceftriaxone, a powerful antibiotic that successfully cleared the bacteria from her nervous system. While Casey and JW both made full recoveries, the emergence of the PORB1A mutation proves this pathogen is actively evolving new ways to migrate through the human body. Humans have been in a direct evolutionary race with this bacteria for nearly a century. In the 1930s, we deployed the first successful treatment using a chemical dye called sulfanilamide. The bacteria produces vitamin B9 to create DNA, a process triggered by a chemical called PABA. Sulfanilamide mimics PABA to jam the machinery. Within a few years, the bacteria responded by overproducing PABA in massive quantities that drowned out the medicine entirely. When penicillin entered mass production in the 1940s, it became the new standard of care. At the time, a small dose of 50,000 units was extremely effective at destroying the infection, but the bacteria slowly learned to survive the drug. By 1972, the dose required to clear a standard infection had skyrocketed to 4.8 million units, nearly a 100-fold increase in just 30 years. The breaking point arrived in 1976. A patient at an Air Force base returned from the Philippines with an infection that refused to die, even after multiple rounds of the maximum penicillin dosage. Scientists discovered the bacteria had evolved a brand new weapon, an enzyme called beta-lactamase. This enzyme specifically targets the beta-lactam ring, the structural core of penicillin, and physically breaks it apart, rendering the antibiotic harmless. Over the next few decades, the bacteria continued to iterate. It changed protein shapes against tetracyclines, stole DNA to resist fluoroquinolones, and pumped azithromycin out of its cells. Our best medical technology has been systematically dismantled by a pathogen that constantly rewrites its own genome to survive our attacks. This leaves us with a very narrow margin of safety. Ceftriaxone is currently the final antibiotic we have that reliably cures this infection. The poor B1A mutation found in Michigan allows the bacteria to spread to the brain, but so far that specific family of bacteria has not yet evolved resistance to ceftriaxone. However, bacteria constantly exchange genetic information with other strains they encounter. They share their most successful survival traits through horizontal gene transfer. The danger is the inevitable moment when the Michigan mutation, the ability to invade the brain, successfully merges with the drug resistance mutations we've seen evolving since the 1940s. If we fail to develop a new class of antibiotics, the convergence of tissue invasion and total drug resistance will turn a routine, curable diagnosis into an untreatable neurological crisis.